Hi, everyone. We are going to begin section 9.2 in the plants unit, and we're going to look at how substances are transported through the phloem. And the phloem is just a little bit different than the xylem, which we finished looking at this week. So today we're going to look at what's called the phloem, which is just another transport vesicle, part of the vascular system inside the plant. Like I said, last week we looked at the xylem, which has substances that move really in one direction here. And today we are going to look at the phloem, which is going to have things move in multiple, two directions, really. So we'll take a look at both of those today. All right, if we're looking at this kind of drawing here, what we can see are really the two types of vascular systems. The xylem, which we see right in here, and the phloem, which we see these tiny little, much smaller than the phloem over here, tiny little vesicles that are going to move substances through the stem, really the root system, and then up into the stem system of our plants. So here again, if we're looking at the phloem, like I said, are the much smaller tubes versus the xylem, which are much larger tubes. Now, the phloem is more towards the outside of the plant, and the xylem is more towards the internal part of the plant, or part of the stem, really. Separating the xylem here from the phloem is a system called the cambium. Here it is down here, cambium, so we get that correct, cambium. Here's another micrograph look. We can see the xylem here are much larger. See these kind of greenish, they have a greenish tint to them, versus the phloem, which are much smaller, kind of a yellow cast to them here. And the cambium is kind of this kind of orange color here that separates the two. So xylem, phloem, cambium. All right, those are the three parts of what we consider to be like the vascular bundle of the plant. Right? So that gives you kind of a good look at what we're talking about. All right, the phloem then, again, it's a tissue, and it's going to include a couple of key pieces here. The first piece is what is called a column, which is the sieve, and this column is separated by some perforations that are the walls, and they are very much an important part of the plant tissue. All right, here is where plant is going to be able to pick up the nutrients that it's going to need, most notably sugar, usually in the form of sucrose that's going to be transported throughout the cell. So when we're talking about transporting organic compounds, that's what we're talking about, sugars, and in some cases, the amino acids also. The plasmodesma, which is an important kind of connection between sieve tubes here, becomes extremely critical when we're looking at the cell wall of the plant. So the cytoplasm, which we know is the liquid portion of the cell, gets connected sieve tube to sieve tube by this plasmodesma here. So we can see one cell to another. All right. The sieve tube walls are here. You see the membranes here, the cell wall followed by the cell membrane. It gives stability and a certain rigidity to the plant in order to allow it to stand upright. So because of this, we have a much higher internal pressure that is associated with this particular stem. Now, the perforated area here is going to reduce some of that resistance, right? Because of the ability of the fluids to move from one sieve cell to the other. All right, if we're looking at, again, we're looking at plants as being photosynthetic. So we know that carbon dioxide comes in from the atmosphere along with water in the root system, and it gets pulled up by the roots against gravity into the upper regions of the cell itself. So we're looking at flow here. Remember, the xylem is bringing water up one direction here in this way, and then the water is moving up and it flows back down. Now, what we will see here is these compounds that get sugar and amino acids that can flow in either direction. That's kind of what makes the difference here between one of the differences between the xylem, which everything is flowing in only one direction, and the phloem, where things can flow in two different directions. Now, a couple of key things to note here. We have what are called, here's our sieve cells, right? Remember, they're connected with the plasmodesma. And we also have down here what are our sink cells. They're going to consume those needed organic compounds, like the sugars. The source of our where our compounds are coming from, right? We're going from source to sink, right? Top to bottom. Our source is going to be any of those tissues that have extra organic compounds. For example, leaves, tap roots, things like that. And then the sink is where the compounds are going to be consumed. Sometimes plant sinks will turn into sources, so and we can kind of go back and forth with that. So we see our leaves and the fruit itself become sources of sugar in photosynthesis, and it gets transported to the phloem, and it can then release and make these carbohydrates in the form of and in the presence of energy, and it can release those to various parts of the plant. Right? We see the fruit, we see root systems, we see leaves, we see stems. So all those things are very active, and it causes the organic compounds, like the sugars, to be sources to the sinks themselves, right? So this is very fluid. So just a couple of questions that you should review, make sure that you're able to answer those in your notebook, and keep that for your portfolio. All right. So that's it on the xylem and the phloem.